record on this. So where was I? Yes, so here we are on our, oh, no, that's my system monitor. Here we are looking at my system monitor. This is better. Okay, so here we are on our training page, rockefelleruniversity.github.io, and here we have everything we will and have covered. Let me know, I've got my air conditioning on. Let me know if it's too loud in the chat. I'll keep an eye on them. But so far we've covered our intro to R, so the very basics of how we can read in data, most importantly, how we can read data from files, how we can manipulate them, and then we saw a little bit about how we can do some plotting. And then hopefully in combination with some of our reproducible R, we may be able to put this into an R markdown, and then we can have reproducible documents containing some of our simple analysis we've done so far. Okay. So we've kind of finished what we're going to learn about how we can do some programming in R. And from this point, and after, this is our last class before the 4th of July. After the 4th of July, we're going to start to jump into how we can apply this to genomics data. But before we get started on looking at how we can do bioinformatics on genomic data, we have this course, which is just called Genomics Data. And it's in here we're going to run through today. And there is no programming in this course. We have or in this little workshop, there is a graphical user interface we use in the second workshop today. But really, this is just to introduce us to the types of data that we're going to come across in genomic analysis. And it's going to include the reference genomes we need. And I'm going to talk a little bit about that at the beginning. But then we're going to talk about sequence data. So this is, of course, mostly about high throughput sequencing. So we're going to talk about the raw data you would get back either from your own sequencing or public places, public repositories, previously published data, and then how that looks like in various summarizations. So, you know, what does that look like once we've aligned it to the genome? What does it look like once we've summarized it to signal within genes, etc. Okay, so today on our class, again, would be genomics data. If you have any questions, again, we've got this Get Help here. Hopefully, reasonably straightforward today. Again, there is no class on Friday. I gave us the day off before the 4th of July, and we have Monday off after, just in case people are traveling. I'm sure we all need a break right now. So let's get started. So today we're going to cover our basics of different genomic data types. So we're going to start by looking at what is a reference genome, and what is this GRC stand for, the Genome Reference Consortium. We're then going to talk about the first types of data you're likely to see if you were doing your own sequencing, so like the rawest data. You're going to get FASTA, which is how we hold reference genomes, and Fastq are unaligned sequences. And then we'll talk about some of the aligned data. So this is when it starts to become useful to us because it's positioned on the genome. We can see where our signal came from. And then we'll talk about BED, very simple way of annotating genomic information, GFF, GTF, essential for RNA-seq, and gene annotation. These formats here we will see, and these are often easy ways of summarizing data, visualizing them in a genome browser. If you work at all with, I think we have a lot of medical and clinical people in this class. If you work at all with variants, structural rearrangements, especially things like cancer, Vcf and Math format will be essential, and you will see a lot of them. For anyone doing single cell, which will be a lot of you if you're doing any genomics, we're going to talk briefly about HDF5. So these are the major file formats I think you'll see in genomics data, and these are all covered in our class, how we can create them, how we can manipulate them and edit them in R. The first thing we'll talk about is reference genomes. So it's a rather trite statement, but the human genome isn't complete. As you'll keep reading, they're trying to sequence centromeres now. We're trying to fill in the gaps in our sequence. Most model organisms are still being regularly updated, including, and we just talked about this in our meeting before, probably somewhere here, Nope. Gosh, I don't have the link to it. But MM9, the mouse genome, oh, here it is. The mouse genome, squeak, squeak, got updated just this year to MM9, MM39. So I think that was updated in February. It was actually updated in January. So updates to what we would consider well-known annotated genomes, these are happening regularly. So be prepared for this. We have reference genomes, but these are updated. Reference genomes, as we're going to discuss them, consist of a mix of known chromosomes. These are our chromosome 1, 2, chromosome X, Y, what you would see in a karyotype image, these kind of chromosome bands. Also have, on top of your standard chromosomes, you have unplaced contigs, which are pieces of DNA. We're not exactly sure what chromosome they are in, but we know that they are together in the genome somewhere. And we may even have stretches of sequence we know are on chromosomes, particular chromosomes, but we don't know where in the chromosome. So all that together, all the DNA sequence we know belongs to a genome. It's called our genome reference assembly. So as I just said, we have major revisions of the assembly. So they're not all quite complete, right? They're all being updated. I mean, they're a long way from where they were 10 years ago, but they are having updates. And major revisions to these assemblies, so what we would call a major revision, will lead to a change in coordinates. And actually, that's what this thing said here, I think, somewhere. This means a change in chromosome coordinates, right? So it's a big deal when we have a major revision, and this is now M39. You can see here, the version before was M38. These are the major versions. So this change in coordinates is a big deal. When this happens, ENCODE or these big projects, they will often freeze on a particular genome version. And when they go to update to a new version, it's a big deal. And they have very carefully planned this action out. So, it requires conversion. There are, and again, that blog will talk about, there are tools to allow you to convert. But we would recommend you just basically start your analysis again. You realign, start from the beginning. The latest assembly of the humans is still GRCH38. There is now, as I just said, GRCH39, GRCM39 for mouse. On top of these major revisions where we have changes in the chromosome coordinates, which means we have sequences being put into our genome, right? Changing, you put in another 100 base pairs, everything shifts 100 base pairs up. We also have these patches, and these can be changes to the major revisions, which don't actually change the coordinates, right? So it could be a change in, this isn't an A at this position, it's now a G, or this stretch of ends unknown in the genome. Perhaps we now know what they are and they can update them. So I think I had, yeah, so the GRC is a collaboration of institutes. I have their page open, we'll hop to it in a second. And their major role across these institutes, including Washington University, NCBI, Ensemble. Their major aim is to maintain the reference genomes for 3 model organisms, mouse, human, and zebrafish. But on top of the GRC, there are big consortiums which manage other genomes, such as Drosophila, mosquitoes, rats, etc. We can just go and look quickly at the GRC. I think I had it here. Here's the Genome Reference Consortium page, and you can see it's made up of many different consortiums. Here's EBI, I said. I thought I said Washington, but maybe I was wrong. NCBI here, Sanger. It's probably the one I was confused with. If you go into NCBI, I think I have it somewhere along here. Maybe not. Somewhere in NCBI, you will find that if we went in there, we would find our genome for our GRC. Let's go there now. We can go to human here. Here we go. So we can see actually now we're on patch 13. We're still GRCH38 major revision, but we're patch 13. So there's been some updates. And we can find information on this here. And we can go and look at the genome if we really want to. Okay. I think Rockefeller, especially, of all the universities I've worked in, has probably the most non-model organisms, or not these three model organisms, or which are used in your science. And actually, I think it's the Chronolab. They're the guys who actually built the colony radar ant genome. Me and Matt were working a little bit to try and annotate this functionally earlier in the year. We have the vertebrate genome project. So we have a vertebrate genome laboratory here at Rockefeller. So we have, they're associated to this massive consortium, the Vertebrate Genome Project. So they're real experts at building genomes as a general. So if you ever wanted to build your own genome and there aren't there in these reference genomes available, you can see that they've got a lot of experience building all these different genomes, sequencing, and they have this technology and some of the bioinformatics to assist you in doing this. And of course, we also have the Bossel Lab. And, you know, most of the modern annotation for mosquito you'll find with them, to be honest, or within their GitHubs and within their papers. So GRC is a great, because it's collected all this, and it's very well organized, that they are putting together these three major model organisms. But other model organisms are maintained, not necessarily by them, but by different organizations such as Vectorbase, such as this Vertebrate Genome Project. Or you can go build your own using the Vertebrate Genome Project. So why do we need a reference genome? Why is it important for us to have this? Primarily is really the second one. I mean, the first one too, they're both absolutely essential. But once we have a model's full genome, you know, and that can be very big and full of areas which we're not necessarily interested in, we can then use our targeted high throughput sequencing and just align that, place that on this model organisms or the genome which has been built already. Okay, so that means that we could just sequence RNA and place this onto the pre-built map of someone else's genome. Okay, so this allows us just to do a lot shallower sequencing and use that map of the genome. Another reason, which is obviously super important, is by having the full reference genome, moving from what we used to have, which was express sequence tags. We knew this was expressed, but we didn't know where in the genome. So now we do know where they're expressed from, and we can start to say things like gene A is close to gene B. Maybe they regulate each other, or they're regulated by the same enhancer, same mechanism. Gene A and gene B are within another feature. Perhaps they're within a TAD, this topologically associated domain, and maybe they're regulated within there. Maybe they're not, so they're regulated by different ways. By having the full linear context, the whole genome, we can start to ask these questions. Okay. So with a reference genome, here we have our reference genome in blue. This is really what we do. I'm going to bring Connie when we come later in the class, and we actually start looking at working at the sequences in R. But for now, typically what you'll do is you'll take your DNA or your cDNA, your copy of your RNA, you copied it into cDNA, you fragment this into small sections of DNA which are broken up from your original DNA. And then to sequence that, we use something like an Illumina sequencer, and we'll sequence usually just the ends of these fragments, okay? But we'll know that we are sequencing, say, these two reads here come from the same fragment, and they will be connected, you know, by that information. Once we have these unaligned sequences, this copy of our fragments, usually just the ends, and then place them onto the genome, this reference saying, you know, this read here matches this bit of sequence, this read here matches this bit of sequence, you know, they're connected. So that's a good place to map this onto the genome. We want to use our reference genome in combination with our unaligned sequences to produce aligned sequences. Okay. So what does a reference genome actually look like? Well, it is just a big text file, you know, several gigabytes long usually. So the human genome, I think, is like 5-6 gigabytes long, the size of it anyway. So it's a reference genome, and it's a collection of contigs, not necessarily the known chromosomes. It can also be these random chromosomes, these unplaced contigs I was telling you about. But a contig is just a stretch of DNA sequence encoded as A, G, C, T, or N. These are our nucleotide bases, N being unknown. So maybe, and quite often you will find huge spans of N in a genome just because we know there's a gap in the sequence, but we don't know what's there. And then every FASTA file for every contig will have a beginning line. So just to say that this contig, what's some information about it? We have this greater than symbol, and then we have some information about the following stretch of sequence. Okay, so this tells us probably some information on numbers from NCBI. These are probably identifiers, and then Homo sapiens, chromosome 1, GRCH38, primary assembly. And then we have the sequence running on chromosome 1. So importantly, a FASTA file has a header line for every contig, and this header line contains this greater than and then some information about the sequence that follows. So for high throughput sequencing formats, we're typically going to get unaligned sequence data generated from these machines like Illumina, and then we're going to want to map these to the genome to produce aligned sequence data. So we're going to go from unaligned sequence files to aligned sequence files. Our unaligned sequence files are going to be usually 99.9% of the time now in Fastq format. So very similar to FASTA format, but now called Fastq. That's our unaligned sequences. Our aligned sequences are going to be in something usually, and again, this used to be more variable, but I would say 99.999% of the time, it's going to be in something called SAM format. Especially modern aligners, modern tools, they're going to produce this type of format, SAM or an equivalent binary version. So what is in a Fastq file? Well, Fastq is FASTA with qualities. So it's going to be very similar to a FASTA file, but it's going to contain additional information, qualities on how confident we are that a sequence at a particular position in our read is truly that base, right? How confident are we that this is an A, C, a C, a C? So we have that too. It's not just the sequence, it's the confidence in the sequence as well. It's made up of 4 lines. Every record in our Fastq is made up of 4 lines. And we have our header line, but this time it starts with an @ symbol, no longer with a greater than symbol. So this is our header line. We then have the actual sequence from the read here. So this is what our Illumina sequencer retrieved from the machine. We have a gap line, which used to have something else in it, but not anymore. And then we have this line here, which is our quality scores encoded as an ASCII value. So they look like ASCII kind of keyboard characters, but these are actually encoded qualities. We'll talk a bit more about that now. So that header line is really useful in a Fastq. And if you're ever in trouble and you can't remember where you sequenced this, or is this genuinely my Fastq, it's your header line which actually contains all this information to help you track back. Okay? And when we submit this to repositories for our publication, often they will scan this header line and it will tell them, oh, it was sequenced on this type of machine, etc. So every line, this @ sign, we have, let's check the chat. There we go. Every Fastq, we have these 4 lines. We have this header line, and we can see it's made up of various kind of blocks of information. This first part before the colon, is our machine name. And if you were to Google this, it's going to be obviously a HiSeq 2000. But there's a whole set of names, and they're not all so easy to read out of the box, but there is maps online, and you can work out exactly the machine type you used. It then tells us the flow cell so like the flow cell lane, the number of the lane within the machine, and then it tells us actually positions within the flow cell. So a flow cell is a 2D plane, and as we'll talk about later, but reads appear or are sequenced along this 2D grid, and this information tells us where it was within that grid. Okay, so this is all really useful information for tracking back not just the machine, but the flow cell and the position. And you can imagine later on, if we had any positional problems in genomics, this part of our flow cell didn't work on this particular machine, we can track it all from here. Last thing we have is our read in a pair. So can it be read 1 or read 2? And this really corresponds to this part here. When we're sequencing either end of a fragment, we have the first read and the second read, and these are paired because we know they came from the same fragment. Okay, so that would be read 1, that would be read 2, read 1, read 2, read 1, read 2. So in this case, we know that this is read 1, so it's got \1. Okay, so then we have this quality line. So this follows the plus, and this is a minus log 10 probability of that sequence base being wrong. Okay, so it's basically a reverse probability of it being wrong. Okay, so this is encoded in ASCII space because, as you can imagine, if we wanted to write out, and this is a probability, it can be a very long number. If we wanted to write this out, every single probability, a long number for every base here, it's going to take up a lot of space in the file. And it's just absolutely impractical. So what we do is we bin our probabilities. So we say from this range of scores to this range of scores, we will call them bin 1, and we'll call this range to this range bin 2, this range to this range bin 3. And then rather than saying 1, 2, 3, they convert that into ASCII numbers, which again, in the computer will save space, right? Now, ASCII is a very simple way of storing information. It's not super useful for us. It'd be great if it was a number, maybe we could more easily read this. But when we start to pass this to our software, use this with these programs downstream in R or other software, it's going to be able to translate these into scores very quickly. So it's not superhuman readable, but computers are going to be able to handle this very fast. And it means that we're not taking up so much space to store the information on how good these sequences are. I haven't got a link to it, but there's a really good Fastq format. There's actually a really surprisingly great Wikipedia page on Fastq format. And here you can see a little bit about how they've encoded this. Hopefully I'm still sharing my screen. You can see they got numbers and they encode this to these bases along here. Again, we don't really need to know this information so much how it's done. It's going to be done under the hood. So when we start working with Fastq, different information is important to us, but it's going to get translated into numbers by the programs we use. But interesting to know, or useful to know, what this line is. Okay, so Fastq is unaligned. We just have sequence information. We have the quality of the sequence, how much we trust each base. We have some information on the machine, but we don't know where in the genome it came from. Okay, so the first or most important format for aligned sequence data, so once we've done our mapping to the genome, is this SAM format. And this was put together by, correct me if I'm wrong, Matt Heng Lee, and he wrote a program, SAM Tools, which we will learn to use from within R. And it was just a very clever thing for him to do, because he put together a format and standardized how we work with sequencing data. Up until this point, different programs which mapped our sequence reads would all produce a different format, and then you would have to convert to keep the information you think is important. SAM format made this all unified. I think every single aligner we're going to show you, and I think most, if not all, modern aligners output SAM format. Okay, so a very standard format for sequence data, recognized by the majority of software because it's kind of won the sequence format war, and genomic browsers. So we're going to cover that in session 2. A lot of these formats are text readable. So all these ones that I'm showing you at the beginning are human readable, sorry, they're text files. There are binary equivalents of them which are compressed and take up less space. Okay. So when I say it's recognized by software and browsers, it is, but there's another format which is the binary version, which is actually better for software and browsers. But I'll explain at the end. So because this is text format, we can actually read this. This is in the binary one. So we can look inside a SAM file and see what it's made of. So the first lines in a SAM file are what are called the SAM header. And it looks just like this if you were to just look at the first few lines of a SAM file. It looks just like this. And it contains information on the rest of the SAM file. So you put this at the beginning, and it can tell you what's coming up in the rest of the SAM file. Importantly, it has this HD tag here, and this is the version number. Okay, so the version number of the SAM tools or the SAM file we have, 1.4. But really usefully, it's also got sorting information. So it tells us here, SO coordinate. It tells me that the rest of the SAM file is sorted by coordinate. I do not need to read the rest of this millions of lines of SAM file to know that, don't worry, it's all in order. It's on the first line. We also have here really useful information, which is our chromosome contig names. So we have chromosome 10, 11, 12, 13, chromosome 1. This is also the order that the chromosomes appear. So you have chromosome 10, and then they're ordered, sort ordered here by coordinates. Chromosome 11, chromosome 12. And then we have the length of the chromosomes. Okay. So this information is useful and used by other programs when we read a SAM file. It's also nice because we can read this and we know the genome by recognizing the lengths. For the actual below this header, we then have the actual information on the aligned sequences. And this contains both alignment information in a SAM file, but also information on the read as well. So we've kind of got the information back that we had in the original Fastq. So just to step through this a bit, we have our Fastq information. So because we use Fastqs to put into the aligners to make these SAMs, we have the read name here still in the SAM file. It's the actual ID for the read. We have the sequence information here. And we also have this Fastq quality score and the encoded ASCII quality score here. And now we have additional information though, because this is now aligned data. So this has been placed onto a reference genome at FASTA format. It's been placed against that now. So we know where it should align to. We can see here the chromosome to which it aligns, right? So that's what was in the header information for the FASTA. So it told us now this is chromosome 2. This read aligns to chromosome 2. The mapping position of the read, and it's actually the 5' end of the read. So it maps to 9 millionth position or 92 millionth position on chromosome 2. And then we have here in green our alignment information. It's actually called the cigar string. And it's another way of encoding information here. Oops, sorry. How is minus log 10 probabilities? That's actually something called base calling. Let me come back to that. I'll just finish this section, Matthew. It's a good question. I will come back to it. Great. It's a good question. It's a good question. So chromosome, where it starts to align, and then how it aligns to the genome. So what we have here is 100M. And this is a way of, again, of encoding information. We have 100 matches in a row. So it's 100 match. Okay? Rather than writing out match 100 times, we just do 100. We also then have some slightly more complex mapping information here. So we have 28M, 1D, 72M. So this is slightly different. It maps perfectly for 28 bases. It then has one deletion from the reference. So there's one read in here which wasn't in the reference. Sorry, one read in the reference which wasn't in the read. And then we have one base in the reference. And then we have another 72 matches. So taken together, this is the chromosome where it starts to align, and then how this read here aligns to the actual genome. I haven't forgotten your question, Matt. I'm going to come back at the end of the SAM section, so just at the end of this slide. Okay, so we also have some more useful information here. So we have here we have the actual mapping position of the read pair. So just to remind us quickly, We have this side of the read. This is the read. This is the pair for that read, right? So these things were mapping or sequencing the same fragment just from either ends. So they are paired. So in this line here, we also know where the paired read mapped to and the insert size, so the distance between these two reads. We have here a set of user-defined tags. So these are actually defined by the aligner. So these can change dependent on the software, but it's just another way of containing information within our SAM file, which might be useful for downstream analysis. So your software will come in, look at AS, find this tag, and then it will parse information from this. The last really nice, useful bit of information, if you're ever looking at a SAM file and you need to just dig in a bit more, is what we have what we have here, which is our bit flag. So if I open up that page, hopefully, I'm not sure where that went to. Okay, maybe I just left my actual page. So another way, again, of encoding information in this aligned read here is using these numbers. And if I type in 147 here and say explain, it can then translate that number and it can tell me that that read was paired. It mapped in a proper pair, so its other read mapped too. It was read reverse strand, so it mapped to the reverse strand, and it was the second in the pair. If I wanted to see what the mate would have looked like. So again, another way of storing information in our SAM file. We just have a number here. We don't need to write out multiple words or something descriptive, just this number. And then we can either go to that web page and translate to see what it is, or in fact, we can just let the software do it for us. Okay. So when we want to pass this to our next bit of software, it will be able to read this very quickly, all these tags, or any of this information. So I think I have another question. Great. So Matt, to be fair, I'm not really going to cover it in this session, but I'll go back to those slides anyway. So yes, so this is actually, so to go from, again, we're going to cover this more for the sequencing, but sequencing is really imaging. So when they add, they do it in cycles such that they will add different colored fluorescent bases to the machine. And they can see, okay, on this particular fragment, we added this read at fluorescent blue. That means the next base in the sequence is A. And then they wash it all off. They put another set of DNA, labeled DNA on. Maybe a green dye flashes up on that. So they can kind of get an idea, okay, green at this time, it was G, yellows and a C or whatever. So they have different colors. The fact is that's not always accurate. And sometimes colors overlap, and sometimes it can't quite get the image correct. So that whole kind of step is called base calling. And there's a program called BCL to Fastq, which I think is the only base caller anyone ever uses for Illumina. So that changes this, what is stacks of images. So if I sequence 100 bases, I have 100 stacks of images. It goes through these images and it works out, okay, at this position in my flow cell, I had a sequence and an A went there. That position was a G, T, and C. But it knows, okay, I think it has a probability of it being any of the bases. And if its probability of it being both an A and a T are kind of high, it starts to downweight this score. So all these quality metrics here, sorry, these quality scores here are from how likely it is a G, C, T, or an A by interpreting that image. So later on in the course, we're going to come back to Fastq and qualities. Connie's going to come and talk through how the Illumina sequencer works. To be fair, really, you'll rarely see anything before Fastq. So base calling won't really worry you. The major thing is when you start to get low qualities, you're either going to have to trim or your aligner is going to have to take care of this for you. It's going to start to say low quality reads, I don't trust so much. So I'm not going to weight it so well in the alignment. That's a brief explanation of that, but we'll come back. For that at the end. Sorry, Carolina, I'm not quite sure that's true for any more logs. And when we're talking about that.
Sorry, that was for the bit flag. I don't know if I understand. Like, is it like a universal marker? Like,
yes, a SAM file is irrespective of the reference.
Okay.
It's a universal truth that, you can take this number, their Picard website, and translate it. And that's the really beautiful thing about SAM format, is it's universal. Mosquito, human, mouse, whatever the reference genome, once it's aligned, this just tells you, the chromosome position, etc. So it's reference genome irrespective. So that's how you get that's how you go from unaligned, and you'll get aligned data. But actually, much of the time, aligned data, BAM files, you will work very quickly to try and move them into something more summarized. Okay, so maybe it will be the number of reads from my attack-seek experiment. I'm looking for open regions in the genome. Number of those reads around transcriptional start sites. I get an idea how open a TSS is. Maybe I want to know the signal across every base pair, how many reads I had stacking up for my RNA-seq data. So these are all very summarized. We don't need all this information on bit flags. We don't actually need the sequence anymore. Once I've accepted it's aligned to that position, I could just simplify that and just say how many reads align to that position. Okay, so there are lots of formats available for that, and that includes BED, our most simple, genomic interval and information. We can store in BED. We have Wiggle and BED graph, kind of two opposing formats, but they do the same thing. And that's for holding genomic intervals again, but usually scores, associated with those intervals. And then we have GFF. So this is how we hold genomic annotation, how we hold gene models. So how your genes are made up of exons is typically held in that format. So BED format's really simple. It's just chromosome, start position of the interval, end position of the interval. So here we have 9 intervals. First one is on this chromosome, starts here, ends here. Second one starts here, ends here. So 9 intervals, usually tab separated. Well, it is officially tab separated between the columns, and you just have chromosome start. Okay. So that's an accepted format, and you can just make that in your text file or export a tab separated file from Excel.
Sorry, Thomas, this would be one, each row would be one of the reads for a particular region, or?
So these are aligned reads.
Yeah, for a particular area, right?
Yeah, so then the BED file doesn't need to be a read, it just needs to be any genomic interval when I want to have some information.
All right, thank you.
Here are my TSSs. I could, show that in this information. I could also simplify my reads to BED files, and people have, but it's a much simpler format, effectively. On top of just a BED file, which may be too simple. We don't have any store information or any name information. We have something called BED6, and this is really common format. You'll see from other places and other databases. Same information as BED3, chromosome start end, but then we will have an identifier. So you can have a name of your TSS, the gene name, or the read name if these were reads, or it could be here just POS1, the peak name. We then have a score to associate to this. And then we have some information on what strand it was, right? So at this point, this was strandless. We didn't know if it was on the positive or negative Watson or Crick strand. Now we can have positive or negative here. Okay. If we wanted strandless, so we don't believe it has a strand, we could just do a dot. Sometimes a star is also accepted. Top of these kind of BED formats, ENCODE, leading the way, has created its own type of formats too. We're not going to go into too much detail. You will see these later on, but they are just extensions of bed format. They're still just text files. These are called narrow peak and broad peak, and they just extend the bed 6 to include p-values and q-values. Okay, so p-value, how significant this peak is, and then the multiple correction. So like, given that we're doing multiple tests, correct this p-value. Okay. That's just another extension we may come across, which is very much like bed. Okay. Another format which is very, very useful in general is this wiggle format and bed graph format, or at least the concept of these are. There is a slightly better binary version we will see later. So a wiggle format really allows you to see signal across the genome. So this is often used if you want to have some score for every position across the genome, maybe conservation, GC content. And it consists of an information line again, here saying the type, but it's saying the chromosome 21, our step size, and we're going to do steps along the genome in 5 base pairs. And then we have a score here. Okay, so from this position on chromosome 21 until this position, we have a score of 40, then we have a score, sorry, 50, then we have a score of 40 until this position, and then this. Okay, for the whole length of the chromosomes, this particular chromosome, and then you would have another line and another chromosome. I think a slightly more sensible format. I'm not sure the sensible is the word I'm looking for there. Definitely a much more, what's the word, compact format, let's stick with that, is bed graph. Okay, so what this has is variable length bins. It's effectively bed 3, so chromosome start end, and then it just has this score column. So when we had bed 6, we had chromosome start end, and we had strand, and we had name, then score. Bed graph is strandless. There is no strand information. It's just chromosome start end score. Hopefully you can see there that it's probably more sensible or compact than a wiggle file. Because a wiggle file, you're going to have to go all the way to the end of the chromosome in steps of 5. And when you have values which are exactly the same, so we have 4 steps here or 5 steps here, which are all 40, I have to write out 5 lines, right? That takes up space. In this one, I can have variable length, so I can go from chromosome 1 from this position until the whole end of the genome, there was no score. That's going to save me a lot of space, rather than writing out every 5 positions 0, another 5 positions 0, 0, 0, 0. It's a much more sensible format. Another thing we're going to cover is genomic annotation. So there are a few different ways to hold genomic annotation, such as gene models. GTF and GFF are the 2 major ones, and they're very similar, so that you almost wouldn't know a difference. And certainly when you start to use the software later on, both can be read in the same way. So this is our GFF3 format, and I think this is the most common. Here we have our chromosome. So it's very much like a bed again. We have chromosome, the contig, the positions are the features on. We have start, end of the feature. We have strand. And then we have information on the feature type. So this is an exon for a gene. We have the source. This came from BLAST. But what's really important about this, oh, I jumped ahead a bit there. What's really important about this GFF format, what it has, which allows it to be so useful later on, is it has this concept of a feature, which is the individual line, but also a meta feature. Right? So this exon is part of this gene. So you can imagine later on, when we need to start to do summarization, give me all the reads which land on this gene. We then want to summarize very quickly over all the exons which make up this gene. Right? When we want to visualize maybe the gene model, as we will do in IGV later on, we want to see how this gene is made. it's going to piece together the exons for the gene using this information. So GFF, GTF format is what allows us to associate lines, these features, to meta features, these like combinations of lines, these groups of lines. The next thing we're going to look at is VCF and Math. And very briefly, we're just going to touch on these. So I said earlier on, VCF is really essential for storing information on things like variants, SNPs, rearrangements. This is all in this variant call format. And again, in its most basic, it's a text file, although often we can compress it into a binary state. And it contains a meta information lines. It contains a header line, so column header line, and then data lines, each containing information about a position in the genome. Okay, so each line is a record in itself, a particular variant, a modification versus the reference. Okay, and what I really like about this VCF format structure is like a SAM file. It has information in this metadata header. And as long as I have this metadata header, I can understand everything going on in the rest of the file. So we have very basically chromosome here, position here. We have an ID, so if it's a known SNP, and I have the reference, so what the actual reference Fastq was, it's a T here, a T here, a G here, and then what the SNP was, what my information or my sequencing told me that I had a C in my particular individual. Right? So here I have at this position for this chromosome, it should be a T, but I have a C. So then we have lots of information in these additional columns, and this is information on our particular SNPs, and this can be variable. But what's really useful is if we look up here, we can see we have the info columns explained. Right, so every tag we have here is explained in here. So I can have the total depth, the DP. I have ancestral allele. It's the T here, so it's like the original allele. And then if I come across here, I have information on individual samples. So I've maybe done this in 5 different samples. Yes, so exactly, Carolina. These are, if we were to go backwards again, starting, once you've done Fastq, you'll always start with, and you'll always go through SAM. The most basic format then is BED. Everything on top of that, really, I see as, everything else I see as an extension of BED, right? This is our most simple. We have chromosome start end, and this is probably one of the first formats we saw in genomic sequencing. This then got extended because we need to have a little bit more information. Okay, let's say we want to know a name for a feature, and then we want to have a score, maybe a strand. Okay, so BED 6 allowed us to cover lots of different types of information. This could even be a gene position or an exon position. Right? But then we got, if it was an exon position, it wasn't quite enough to allow us to tie it to genes. So there is a better format for then tying exons to genes, and that would be GFF. Again, a BED file or a BED 6 file could contain, a name, a strand, and scores. But actually, it's easier here to have a BED graph. We don't necessarily need the name. We don't necessarily need the strand. So they are all very similar formats, just built now on top of that concept of just chromosome start end, kind of building on 4 particular problems. A VCF is good for variants, and it's become almost the winning format. You will see it a lot. You could still put the information in a BED 6 probably. Yeah, exactly. Yeah. GFF, you could put the same information in a BED 6 file, combination, but GFFs are recognized now as a format, and when we go past them to software, we've got software waiting for GFF files. So we have to kind of follow the trends in a way, and these are the types of files we're going to see a lot of. Okay, so that was VCF, and again, just to reiterate what we just said, chromosome, start position, an ID. It's very similar to BED, but it's now really tailored for variant information. Strip out what you don't need and leave in what you do need. Most importantly, VCFs are expected to have this metadata beginning, right? So that anyone who comes to it, you have the expectation, I can explain what all these columns mean. So just slight extension of VCF is a mutation annotation format, and this is really an aggregated mutation information from your VCFs. You won't see this quite as much as you will see VCFs, but it's also more gene focused. And what you can see here is you have the annotation for the gene that the SNP was on. So telling us now it's this gene, this ID. We will get some classification of what type of mutation it is too. So here in this case, we get like a splice site, a missense mutation, or maybe disappointingly a silent mutation that didn't do anything. So this is often what you'll see when you're starting to assess what is the impact of my SNPs on my gene expression or potential impact. Okay, so I've talked through all these different formats. I'm sorry, I'm just running out of time a bit. We've talked through all these different formats. Sorry. Is the reference genome specific to a strain? Oh, yeah, that's a really good question. Can I come back to that at the end, Abbas? That's a great question. It's actually a combination of strains, deliberately. It's a really good question. You can actually, if you go onto that page, NCBI, you can actually investigate, see what they've done to try and avoid the problem of having bias to one strain. Yeah, but a really good question. We'll hop on that page in the break. Okay, so then, I've been saying that all these are the standard formats, and they are absolutely the standard formats, but they are the text readable, the text version, human readable versions of these formats, which is brilliant because we can go look at them. And I always feel much better if I can open a file and actually look at it. This is the line which is causing me a problem. This is the line which is really interesting, this particular feature. There's nothing wrong. It makes sense in a text file. But that's not great for software. It's not great for space, and it's not really great for software. Actually, text files are terrible for scanning through. You can easily save or make these files much more accessible to software. And using this in combination with something called indexing, we can actually allow us to access parts of these files without having to read the whole file. Okay, and I can tell you some of these BAM files are hundreds of millions of reads long. Some of the ones you will sequence as well. Reading 90 million reads to get to my 90 million and want to read can be a real pain. So these formats allow us to deal with the data much more efficiently, and they allow the programs we use to deal with it. Whereas we have SAM files, and SAM is exactly the same information as a BAM file. We also have this BAM file, so sequence alignment map, binary alignment map. And with a BAM file, when we produce it, you'll often get a .bai file. We will show you how to make these, and we will show you how to align data, but you can also download these as well. So if you download them, you get a .bam and a .bai. So it's binary alignment map, binary alignment index. This is the index of this file. Wiggle and bed graph, both can get turned into something called a big wig. Okay. So this is a UCSC thing. It's either ends in a .bw or a .bigwig. These are, again, compressed versions, which are very quick to read. They have indexes built into them so that we don't need a separate index. But when we go load this in software, we don't need to read all the way from chromosome 1 to chromosome 20 to visualize a bit of chromosome 21. It will just read the bit it needs. Equivalent to bigwig is big bed, but not so much used, because really the winner, I think, for compressing and indexing and retrieval from bed, and this really comes back to your question, Caroline, as well. These files are very similar because they all can be compressed into basically the same type of file, which is this gzipped version of themselves. and they have a .tbi index. So this is different from this. It's called a tabix indexing. And it's called block compressing. But often we will get BED, BCF, and GFF files. They will be gzipped and they will be accompanied by this .tbi. This just means that, like with SAM, the BAM files here or bigwigs, if I've got a million different SNPs, I don't need to read them all in to get to my 1,000,001. I can just hop to 1,000,001. So the last thing we're going to talk about very quickly before we end the session is this new format that came up more recently, and this is HDF5 and Loop. And we're just going to touch on it because we're going to really use it later on. But the results from many experimental assays are summarized into combinations of tables. So often you will have a few different tables which contain all the information to describe your experiment. So one may be, if you're doing RNA-seq, for instance, you may have gene expression measurements. So the rows are genes, your columns are samples. You may have then additional metadata on your samples, which you want to store next to your information on your expression values. You may have additional information on your genes, right? Annotation, this gene is this function, or this gene is this particular model. And you may also have multiple copies of these measurements, but even tables describing the transformations you're performing. So historically, and me too, you would contain this type of information, or if you wanted to give this information to someone, you may have either one mega sheet, but if you're a bit friendlier, you may give them separate sheets, one containing your expression data, one with your group info, and then one with your gene info. Okay. That's how you would hold that in maybe Excel. I mean, I think naturally, if you wanted to hold a matrix, a data frame, and another data frame, you'll do that in the list in R, right? But holding all this data in lists, especially if it's really big, having it in Excel is unwieldy. Really, we want with bigger data formats, and single cell has really pushed this. We want something which is easier to allow us to share, both across different computers, clearly, but different programming languages. I may want to do a little bit of Python. I may want to use someone else's software and come back to R. We want to store and connect different data types, like data frames, matrices, all in one environment, allow us to move between them easily, and it needs to be fast and memory efficient. Right? So I can tell you, even saving some of these files, like single cell files, they're just gigabytes big, and you would be kilobytes before. Now they've just got so big, you probably can't even open them in Excel. Excel's actually got a lot better recently, but it would struggle. It would slow your computer down. So we need fast. We don't want to wait around, but we also need really memory efficient. And in the same way I was telling you this concept of, I don't need to read all the data in order to get to this bit of data. Formats have been developed to allow us to do this more generally. Okay, and one particularly fantastic format is this hierarchical data format, HDF5, produced by the HDF5 group. You can go to their website if you want to read more about this. But effectively, it's a format, it's a type of data format, an associated set of libraries. So they've written a lot of low-level C to allow us to interact with this. This can work on all computers. Lots of different programs, Python, R, interact with it. What's nice about it is it actually describes itself. So these different columns, these different sheets, kind of similar to what we had here, this metadata information, describe what's coming up in the rest of the file. HDF5 more generally extrapolates that. So it allows the file to describe what's coming up. What do I have within me? It's fast, very, very fast, and efficient memory I/O. You can read parts of it. You can actually operate and do calculations on parts of it and do it in blocks. So that makes it very suitable for large data sets and their associated metadata. One place we're going to see this straight away is in single cell. Right? So single cell, I showed you these BAMs, I showed you these bigwigs and all this. Single cell is quite sparse, so bigwigs often aren't particularly useful. There are ways that they are useful. But one of the first things you will see is this type of format. You won't even see the band. Like a lot of people will just get this type of format, and then you will start to work from this. So this really is a very important format if you're working with single cell. Single cell clearly and single cell attack, single cell RNA-seq, both of them have this large matrix, cells, 5,000, 10,000 cells by 30,000 genes. So very big matrices, and they have lots of information associated to the columns. So your cells have lots of information on them. Your genes have lots of information on. All that is stored in this HDF5-like format called a loom file. And a loom file is just an extension of HDF5, implemented in Python, but importantly for us, we're going to be able to interact with it in Bioconductor, which means we can use both types of tools. We could use a bit of Python, then a bit of Bioconductor. And what's really great is it is incredibly memory efficient. And in combination with some of R's clever tricks they're doing recently to make things more memory efficient, this is going to allow us to do big data sets before our computer blows up. It's really going to be able to allow us to do proper single cell analysis inside R. So that's the end of this current session. I'm going to end it there. I have no exercises for the session. What I tell people when we get to the end of this session is to start to try and look into your own lab's data or your own projects and start to see if you can find some data for your lab and for your experiment, and see if you can find them in these formats. Do they have BAM formats? Do you have Fastq available to you? In our next session, I'm going to quickly run through this genomic data repositories. So this is just going to tell us then, if you can think about what type of data your lab has. I'm going to show you where you can go grab it from. And then I'm going to introduce you very briefly to this wonderful GUI, IGV. So this will be, again, no programming today. Sorry, my computer's. But this is a nice tool which just allows us to navigate data in a genome browser, in a nice linear genome. And we're going to hopefully already be able to start to use it with public data. So we can start to take other people's data, maybe your own lab's data from previous publications, start to review it inside IGV. Okay, so there's no exercise. If you have any questions, maybe we still had some there. I'm going to find a good reference for you, Abbas, about the mouse genome. I know I came across it earlier on. It's very similar to what I said, but I will get you the proper information now. If you have any questions, I am around in the break, but as I say, no exercises. We will pick this up at 3 o'clock. Please remember, everyone here, we don't have class on Friday, but I'm around on Friday. I may not be in the office, but if you have questions, just drop them in and we can answer them. I'll speak to you in an hour. Morning. Then share my screen. Okay. So we were in genomic data. I'm just going to go back for one point. Let me refresh my memory on this. Yes. So one point, I just want to go back to the, because we had two questions on it, and they're both good questions. Asking a little bit more about, let's not do it like that. Let's do it properly. Fastq quality. Okay, so what are these quality lines? How are they derived? So I just picked up an image, which I think will much better describe this than I am doing here in words. But just to reiterate what I said, every Fastq has 4 lines. So sorry, every Fastq entry, every read has 4 lines. So we have a header, sequence information, our plus line, and our quality scores. So then these quality scores are the -log10 probability of that sequence base being called wrong. Okay, so the higher the score behind this ASCII code, the more likely, like the the more confident we are in that base being correct. And really it's derived from this picture here, this one here. So this is a really lovely image of like how this is done. So if we look along the bottom, we have our colors, and I was saying that we have different fluorescence for different colors. And this is working along a sequencing cycle going from the beginning of our read that we're sequencing to the end. And we can see what we have here is the intensity of the fluorescence when we are looking at that particular spot on the flow cell, that particular sequence. And at the beginning, we have quite black only, so we're pretty confident it's a G, and then we're confident it's an A, and T, T. But as we move along, you can see this confidence here. This is our Fred score, goes up and down depending on how confident we are. We get to a point along here where we start to have a lot of background, right? We have a lot of blue signal, okay? But we have blue signal and other signal underneath it. And this kind of noise, then we have some red, and then we have this other noise coming up here. All this leads to this problem of not being, this is actually an A, it could be a blue, it could be a C too. And this is what you see reflected in the Fred score. So when the optics reading, you know, the fluorescence coming off the machine, when it's no longer confident, oh, I put a C there because I don't see enough blue, or I see blue and green, this is reflected in your Fred score. And this ASCII code here is just the way that we translate a bin, a range of probabilities, into something we can encode. Okay. So what you're really seeing here is a reflection of the sequencer just not able to tell, be confident it was an A, a G, a C, or a T, usually by the colors overlapping or some fluorescent background. So that was the reference genomes and genomics data section. Let me put my chat again. Okay, perfect. We can look now just quickly at this short section here. So we should have covered most of the genomic data types we're going to handle. And as we go through the rest of these courses, we are going to come across them. So we're going to learn, first off in our next session, how to just look at them in an IGV, a linear genome browser. And then we're going to look how we can read them into R. We have special structures, like we have lists and data frames. We have special structures to handle bed files, BAM files, etc. But what's really important is actually getting a hold of some high throughput sequencing data. And as some of you have mentioned in the chat, you already have data and you've come to this course hoping, and I think we can process or analyze your data following the methods that we have in class. Okay. So you're going to have some of your own material, maybe you sequence. The other source of material you'll get is often you'll be, you know, if you get handy with your R and bioinformatics, you may have your collaborators come to you too. So they might come to you and say, oh, I saw the work you did on your own material and your own data. How about you process mine? Probably more likely, they may share it with you. They've already processed it, but they will give you the raw data. And they will say, yeah, sure, go and investigate this data versus yours. Something we do a lot as a bioinformatics team, and I hope you will do a lot in your own analysis, is even if you have your own data, or if you don't, there's so much data available in public repositories, and it's part of publishing scientific journals, or at least biological scientific journals. You have to make your data available. And high throughput sequencing data, as we will see now, is very strict on this. So if someone's written a great publication and they've created some really interesting data that you want to get your hold on, you want to get your hands on, you should find it in some of these public repositories. It's a requirement of the funding. Most fundings, most journals. There's always a journal which manages to escape putting their data in public repositories. So for high throughput sequencing data, which is what this course is really concentrating on, we have three, I would say. There is a Japanese one I'm not going to cover, and that isn't because it's not awesome. It's just I don't use it myself. But the three major repositories which we will use are GEO, ENA, and SRA. So two of these being the American repositories, GEO, and SRA, and then the European equivalent, EMA. So GEO holds different types of biological data sets. There's their gene expression, right? But this is from different ways that we've measured gene expression. It is probably the most popular place to submit publications. So if you are doing your own study, assuming it's not some protected patient data, it's going to end up going through GEO. You will probably submit it, maybe you or your bioinformatician or your collaborator, they will submit it to GEO. And it's this popularity really which makes GEO also so useful, because people are all submitting their data there. It's the place where you can first find data associated to a paper. And it does a good job of capturing metadata, processed files. Not such a great job of capturing processed files, but it does capture the raw data, or at least it tells you where the raw data is. Importantly, it captures a ton of metadata. Not always the same metadata from different studies, but there is a lot of information in GEO. The slight problem with GEO is that it wasn't built for high throughput sequencing data. This gene expression part is true, but it's not, high throughput sequencing gene expression omnibus. It was really built for microarrays, maybe even predating that. So because of that, they've had to change some of the internals as they've gone along, and it's perhaps not tailored for high throughput sequencing data, right? So this is what gene expression omnibus looks like. I had it open somewhere, so I wanted to just go search for something. Let's try here. So here's the gene expression omnibus. If I want to search, how do I search? Here. I can free text search. I can go Mac, ChIP-seq, CH12. So I'm looking for a particular type of data, particular transcription factor, and a particular, so cell line here. It's got 7 results. I can go in and we can see we have various studies here. And in fact, I just want to select the second one to show you a little bit about what's inside. So we have status is important. Most of the data sets you're going to see are going to be public. When you submit your own data set, it will be private for a bit. And then it has information describing the experiment, a summary, very free text, right? There's no rules about what people can put in here. The overall design. These are more rule-based. There's always got to be someone who can be contacted by GEO. So if you ever have a problem with someone else's entry, you have a contact, usually either the PI, so usually the last author, and quite often you'll have a paper associated to it too. Often GEOs are filled up before you've published your paper, sorry. So quite often the publication isn't actually right. It's not the right place for it, but in this case it does match. And then if we look here, we can see we have lots of data sets within this GEO. This is a composition of other data sets. We can go find the one I was interested in, CH12. I was interested in MIC. That will do. And if I go in there now, I can look at that particular data set. And in here, I have some more information. I have the source name, some characteristics. Often I will have something on the protocol, but there's not much information here. Library source, library selection, treatment protocol, none. Extraction protocol, link to somewhere else. That might be worth following. Okay. Oh, I guess they've linked to a description on another page. So there's lots of information here. What is slightly different about GEO to these other ones I'm going to show is, although it doesn't hold directly the raw data, what it does have is some process data. So here we can see something I've already mentioned, a narrow peak file. We know that this is, let's download it, why not? We know that this is very similar to a bed file, but with p-values for the enrichment. This is a ChIP-seq. And then another file, hopefully we recognize, a bigwig. So these are processed data files. This is summarized position. This is going to be score. There's no raw data there. Okay, that's because raw data for GEO now is held somewhere else. So even though you may submit your data to GEO, will go there and do the submission. It will pass it to its partner, which is the short read archive. So this is SRA. So SRA, by its very name, is built for short reads. The Fastq data we get from Illumina, actually, they have their own problem now because short read isn't really going to cover enough. There are long read technologies. If we're lucky, we'll be able to get Olivier to come in and give a little intro on that. But these include things like PacBio. SRA is trying to now include them too. So they have a similar problem. They're updating. They want to be something they didn't originally set out to be. But who doesn't want to be that? SRA stores, as it says, as I said, it's particular for sequencing. It is where we store the raw data for GEO. So all the information, when we try and find raw data in GEO, it's going to point us to SRA. And actually, I didn't click on it, but there was a link there in GEO to go to SRA. SRA has this fundamental problem. I think perhaps there are ways around it, but it doesn't make it easy. But then it has its own format, SRA format. And remember, SRA probably was around before these formats got formalized. So SRA format, you can't really use it directly with other software, but they have a toolkit called SRA Toolkit, and actually allows you to convert SRA format into Fastq. And Fastq is our unaligned sequence read. It is the raw data, and if we want to start again and reanalyze someone's project, if we want to say map to a new version of the genome, we want that raw data. So to get it from SRA, we're either going to have to, we are going to have to somehow install SRA Toolkit and use that to extract it for us. So let's go look at SRA. SRA. So here's an extension. It's actually, I've gone to SRA. Let's go back a page. Sorry. NCBI. SRA. It's all in NCBI. If I search that again, it's here. So this looks a little bit like. Sorry, where did I go there? Sorry, I click on the run. So you can see here, sorry, we have the run, and this tells me the number of bases covered, the size of the file, and I can click in here. Now I can start to step through and download the data. But if I come to download, say Fastq here, either Fastq, sorry, download. Yeah, getting SRA reads is going to talk about SRA Toolkit. So it's not particularly useful. There are some nice systems in here to upload to go from SRA to Amazon Cloud. But I think just to grab data, downloading SRA Toolkit, having to step through the complexities of going from GEO to SRA, and then from here, having to forward through to here, and then work out the toolkit, it is actually kind of complex. I think largely unnecessary. Okay, so I prefer the place I get my data from, and we're going to show you, I think, how you can do this in R programmatically too. The place I get most of my data from, and it's not just because I'm European, is from ENA. Okay, so this is the European High Throughput Sequencing Repository. It is effectively a mirror of SRA. I think it's a few weeks, sometimes behind. So you may find new published data you can't quite get on ENA yet. But it also stores raw data formats for high throughput sequencing, but it doesn't have SRA format. It has Fastq format. So we can go to ENA. Oh, this is the bit I wanted to show you from SRA, sorry. I'll come back to that later. So this is ENA. Let me start that again. So let me just come back to the home page. So this is what ENA looks like. The problem with ENA is it's not quite so friendly with free text search. It won't even let me type that in. Nick, Mel CH12. Oh, I can't type, I'm sorry. Oh, we got something there, but it's not actually the one I want. But it does have, you can search using SRA IDs or GEO IDs, GSM IDs, and you will get very similar information passed across. What's really great about ENA is it has a very simple table. I can download it as a TSV. But in here, I have the links to the Fastq. So if I want to go get this Fastq, my stepwise process is I will go into GEO and I will search for it. I've lost GEO now. But I will search for it in GEO. I will get this SRX number, and then I will come to ENA and I will find it here. And often I will just download the table because the table has these direct links listed in it. And I can just go download them. So I find that combination of these, GEO to ENA, is usually the way to go on this. But remember, SRA, GEO itself doesn't hold any of the sequencing data. It's actually SRA, which is now specially built to use this. Another place worth mentioning, and we're going to use this repository of data throughout the course because it contains both processed and unprocessed data, is the ENCODES Consortium's portal. So we have a link here. You can also get ENCODE, as you'll see, ENCODE data from various other places. So other people put them on their own repositories and their own consortium sites. But it's because it has processed and unprocessed, it's particularly useful to us. So we can grab the raw data and we can analyze it how we would like to analyze it in class and see how we do comparing versus their processed data. Do we get similar results? If we don't, do we understand why? So I'm not sure if I have a link to the portal, but I'm going to go there anyway now. Oh, there it is. So here we have ENCODE, and it's got data and a data experiment search. This is often where I will come. And you can see it's much more arranged, much more user-friendly, really, to get out particular types of sequencing data. And you've got different marks you might be going for, different organisms. We had someone who was interested in Drosophila earlier on. I saw Drosophila there. And then I could go in and decide I want to look at total RNA-seq. And then once I found a data set I like, every data set, every experiment has its own page in ENCODE. It's quite clear here. Yes, actually, this data hasn't been processed or analyzed yet for some reason. But any data associated to an experiment, we can find links to the files here. In fact, I think often if you click on it, it will take you through. It's just got more and more information on this particular data. That wasn't the most complete data set. So let me try. Let's try the CH12, Nick. Yeah, Nick. ChIP-seq. Yeah. You can see here, so it really is an incredible amount of data. Tells you how every file was created. And then if we come in here, we can actually get the raw data, raw sequencing data, these FASTQs we've been looking at and discussing the quality. And then all these other files we've been looking at this, in our other session. We have our big bed, our big wig, our narrow peak, and our BAM files. Okay, so actually I've downloaded a few of these now for our next session. So that, there's a never-ending list of repositories. So you can go and review what the latest set are yourself. But those were the ones which cover most of the raw data. The other types of repositories that I find particularly interesting are the ones which have taken huge sets of data and they have run them through standardized pipelines for you. Right? So that's going to allow you to compare one publication maybe to another publication, at least in the same way that they've been processed in the same way against the same gene models, aligned to the same genome. A really good one for that is this Recount2 database. That's fantastic. And if you go there, you'll find it's processing lots of publications using its standardized pipelines so that you can kind of compare across. And other ones which are great, ImGen, which I use a lot. This is immunological signatures, gene expression, and things like the Expression Atlas, which contains information on the makeup of lots of tissues, and you can just download pre-computed expression matrices from here. Okay. Final thing I wanted to talk about was, so that's how we can actually go get some process data or some raw data, and we could use that now in our following lectures to go work out exactly what we wanted to do, or sorry, to give us some nice example data to really see exactly what our methods we're going to use, or exactly what they're going to produce, and we can compare them versus the results in these papers. That's great, but we're going to need another component. So we have the raw Fastq in lots of cases here. We actually need the reference data. So we need to get our FASTA and the gene models, and any annotation we want to put to these genes, function annotation, we want to get that too. So reference genomes are available from many locations, and it can be quite disparate. For the non-model organisms, remember major revisions, get rid of one of these eyes here. Change locations, minor revisions, usually just update the annotation sequence. So you need to keep an eye on this. And really you want one place perhaps where you can record where you downloaded your reference FASTA, where you downloaded your gene models. So genome sequence, the G reference FASTA, gene models is GFF and GTF. Like I was saying, you can get these from lots of different places. I recommend, if you want to follow along and do some analysis now, iGenomes is a really great source of all of these annotations we need. And what they've done is, for lots of model organisms, and you can probably go through their overview here. Actually, we can just step in quickly. But they're going to give us the FASTA files we need, the GTFs we need, but also some additional things like annotation, some of the indexes we may require later on for alignment. It's pre-computed them for us. So all of this will be available, and we can download just by clicking, I'm interested, say, in Drosophila. If I want to get the ensemble build, there's an NCI build, there's a UCSC build. I'm going to talk a little bit more, because these are all the same version, just slightly different annotation styles, the way they name chromosomes, for instance. But I could just go here, click that, and it's going to download a zip or a tar file containing FASTA, the gene models, everything I would need. I'm going to stop it though. So I can tell you, once you download that, that is probably 40, 50 gigs. It's a lot of data to download. It may be smaller, but you're going to unzip it and it's going to be huge. But iGenomes is a really good source. If you have any problems, if you're working on a strange genome, you want to follow along, just contact us, and we can try and help you find the place to get the information from. But iGenomes, as I just showed you, it's a really great place because it covers most of the genomes you will work with, you'll find here, and you can get everything you need in one zip. Once you've got that, just store that, keep that safe, and that's all the annotation you would need. So that's the end of genome data. So we know a little bit about what type of files we may handle. And we know basically we're going to go to GEO and then ENA if we want to download the Fastq or encode other websites. They may contain some process data. So it's worth looking around. If I'm working on MIC, I will go to encode and just see what data is available to me, and I can just start to review it straight away. Nicely leads us to our next course, which I've only got really half an hour, maybe I'll be a little bit over in this next session to run through, and that is IGV. So what is IGV? We're going to run through it anyway. IGV is a genome browser, right? So genome browsers allow you to review the linear genome and any data that maybe you've aligned to it in the context of that linear genome. Okay, so I can take my bigwig or my bed file of positions, and I can load them up, and I can review them by scanning across the genome from the beginning of chromosomes to the end of chromosomes. Okay. It's going to be even more apparent when we start looking at it, what it is. Okay, so this allows us to visualize genomics data, yes. What is slightly different about genome browsers like IGV is that maybe some of you are familiar with UCSC. UCSC is UCSC browser, not just the university. That is a web-based genome browser, so you can upload your data there, and a web page you can scan across and look at the data. IGV doesn't work like that. IGV you download and it runs on your computer, or at least the desktop version we're looking at. And that has some advantages and disadvantages when you're going to compare IGV to things like UCSC, in that because it's running on your computer, it's going to consume some memory on your computer. If you're going to try and load 1,000 files and review them in your desktop, you better have a very fast desktop to do it. But that said, because of these types of files we're going to look at, BAM files, indexed bed files, we know that we can just read parts of these files into memory. We don't need to read the whole thing because they have these indexes. So we should be able to handle most of what you need to do to visualize your data locally on IGV. That said, UCSC is really popular. We don't cover it in this course, but it's just a preference, personal preference for myself. IGV was really put together by the Broad Institute. There is something very similar called IGB, Integrated Genome Browser. This is Integrated Genome Viewer. It's just the one we've chosen. So IGV runs locally. It is a Java program and it is available from the Broad Institute. You can just go to this website and you should be able to download it. I'm going to step through these slides and then we just go and actually have a look. Maybe we just go have a look rather than step through the slides. Okay, so I think things have updated a bit, but if you want to get Mac, you can download this one, the one for Windows, the one for Linux. What is quite nice is that if you ever find yourself in trouble that you aren't seeing your data in the same way you used to, maybe it's because IGV has been updated, maybe it's changed the way it visualizes things. It also has archive versions. So you can also go back and get older versions of IGV. So as I'm concerned, that hasn't ever happened. The only thing is that we have some software which uses features of IGV. Maybe these change or the way to interact with them changed, that kind of broke. If you want to get IGV though, the major way is just to click here and it will download both Java, so it's the dependency for IGV and IGV, and you should be just good to go. If we have problems, I'm going to be around at the end and we can work through installing it. Okay, so then this is tricky for me because I'm on a laptop and I've got my slides and my IGV behind here. So I'm going to talk through some slides. Just to make sure I'm still recording. Am I definitely recording? I'm going to talk through some slides and then I'm going to hop into the GUI just to show you it actually in action. So this is what IGV looks like. When you load it, it looks like this. It's actually made up of 4 panels. You have a sample information panel on the left here. You have a genome navigation panel that really covers this top section. And then you have 2 very similar panels. You have what is a data panel. So the data you're going to load should pop up in here. And any attributes for the data, and that includes things like genes associated to the genome, will pop up here. Also, bed files tend to pop up in this bottom section. Depending on what you're on, if you're on Windows, I think it keeps it all together. If you're on Mac, it's going to put your file window at the top of your bigger screen. You should have these general tabs available to you. File, so that's how we're going to load data, sample information. We have genomes, load and manage genomes there. So that's how they have genomes built in, but if you need new genomes, you use this. View, this is how we're going to control how our tracks are displayed. We might want to change font sizes, etc. We can change that all in view. Tracks is just for allowing us to arrange our tracks, filter them, sort them. Regions is just ways of us creating bookmarking regions, creating region lists. And then we have tools which allow us to interact with IGV. Actually, I think they've taken the interaction out with bed tools, which is a little bit of a shame. I might be wrong. It looks like they have. So moving around IGV, we can use the cytoband selection zooming. I'm going to show you that now. We can scroll, or we can just select a region of interest. And if we look here, Actually, I'll show you in the real thing. So this is moving around genomes and zooming and scrolling and jumping. So let me show you that in IGV. I should have IGV here. I'm going to share my whole screen though. I think, you know, parts of it might be in my window outside the share. So I'm sorry for this. So I've already loaded some stuff into IGV. Let me re-clear that new session. So this is what IGV looks basic. Okay, so I can see all the chromosomes along the top. Yeah, so I got chromosome 1, and they're all kind of proportionally sized, I guess. I got my genes down here. This is my data panel. This is my attribute panel. This is my sample information panel. This is my kind of moving navigating panel. My system drop downs are all here. If I want to move around, I can just hop to a gene of interest. I can just say I want to look at actin beta. And it takes me into actin beta. And this is our gene model for the gene here. So these are our exons. And this has all just come great from installing IGV. It's got MM10 built in. I can also navigate with this zoom. So I've got my zoom here, go in, go out. I can select chromosomes, etc. Or really usefully is if I see a feature I like, if I'm looking at my data, I want to zoom in on a particular feature, I can just navigate like that. And I think you can even, if I put my cursor there, I can use my keys to navigate up and down, left and right, sorry. Computer game. Okay. So that's how you can very easily navigate around IGV in the windows. Bookmarking regions of interest. So we can also, once we've been moving around and we find something we're particularly interested in, we might want to bookmark this, say this region, and come back to it later. So we can have bookmarks for regions in IGV very simply, either using a way of bookmarking the visible window or selecting the region within the window that we want to bookmark. Okay. So we're going to use the drop-down region navigator, and I can just select and bookmark a region. Okay, I think both of these are things I will show you inside IGV itself. So I think the first way we do it, if I just click that and zoom out. Oh no, I have to do it like this. So that bookmarked my region here. So let's do that again. Let's say I want to bookmark the beginning of this gene. I'm going to zoom in over here. I can go click here, define a region of interest. I just define from there to there. Now I've got my bookmarked region here. Region gene lists. Sorry, close. Region navigator. And then my regions now are here. Okay. Scanning through to see what we want to talk about. Really nice, and what we do use this for with these bookmarked regions. Once I've scanned a region I'm particularly interested in, I can then right-click it, and I can add do things like edit the description, so I can give this an actual description, test region. I could copy the sequence for a region. So a lot of people find that really useful for primers. You've got this region, I just want to be able to extract some primers from it. I can select that, I can copy the sequence, and now if I had a R session open, which I don't, let me open that. Hey Siri, can you open R Studio? You can then just copy and paste straight out of here into. It's copied the sequence for me there. I can also directly black the sequence. So I can use it to kind of search for similar sequences across the genome. And then I can just go and look to see. So it kind of mapped here. That sequence mapped a little bit here. And then it had a gap mapped over here. So just from that region of interest, if I want to find, once I've bookmarked it, I can just right click on it and start to either search for similar sequences or copy the sequence out. There should also be now, sorry. in my region of interest navigator here, I've got 2 regions. And you can see I've got my second region with my description here, and if I want to clear this thing behind it, if I ever want to jump back to my regions of interest, I can, or bookmark regions, I can just do this, so I can jump back to the first one. Great. Actually, you kind of saw here, but I had a predefined resolution in IGV. Once we've zoomed in enough, the sequence underneath where we're looking will start to pop up. So we'll start to see the actual sequence on the forward strand. And then we'll also start to see the translation of that sequence in different codon steps. And if we want to, we can actually click on the right of this and we can change things like the strand. So if we know a gene's on the other strand, we might want to flip the strand, or we can choose more obscure translation tables. So if we're working in a slightly weird genome, we may have a translation table that CTC goes to a particular amino acid in our organism. I want to show you that in kind of blocks rather than keep skipping back and forward. When you do have this region of interest, you can also create lists of regions. Okay, and actually inside IGV, we have some pre-created interesting regions for us. And we can go have a look at them now in IGV. So if we close that now, just to show you, if we zoom in enough, although nothing's visible yet, we can start to see the sequence pop up now. So it's trying to make it reasonable for us to look at. If we carry on zooming in, these little blocks of color will give way to the actual color they represent. So now I can look. Maybe I would want to see if I could find, something simple like a start site or a particular motif I'm interested in. Again, I could bookmark this region, delete that region. I can just bookmark this region now. Sorry. Bookmark this region now. Maybe I just want to extract the sequence from there. And again, I can flip the strand. And now it's going the other way. So I'm going to get the complement, or I could do my translation. Different types of translation tables we use. I think I've got to zoom in a bit more to get the actual translation table. Show translation, sorry. One thing there is gene lists. So we've already started our kind of region navigator. We started making our own kind of gene list. But built into IGV, we have gene lists. So we could import our own. I'm sorry. If we look here, we're going to come across, but we have things like bed file. So we can import a list of regions as bed files. This is basically a bed file, right? Chromosome start, end. Or it had something there of GMT. We'll come across that in a bit. But also there are built into IGV some of their own regions. We're going to come look at this later on, but let's just choose one with a few genes. And then it will give me this split view. So it allows me not just to look at one gene, but now I can look at like multiple genes at the same time. And review here, I don't have any data loaded yet, but I can review the structure of the gene perhaps. And then once I've loaded data on, I can review all these genes at the same time. If I were to zoom into one of these genes, I can click on this and it will take me to that particular gene. I can go back to that gene, go back. If I want to escape this, I can just press home. Oh, it didn't work, that's fine. Maybe I click here. Oh, wait, now I've frozen IGV. Try that, now I'm back. Okay. The most important thing we really want to do in IGV is actually load either our data. We don't want to just review the genome. We want to actually load data into it, either from ourselves or places like ENCODE. Maybe we've downloaded them. So the first thing we need to just actually get the genome we want to align to. We have aligned to, we want to visualize against. IGV has many genomes built in. And if you don't find it in the immediate dropdown, you can actually add to there from the server IGV has. So it has a big server and it contains lots of the genomes. Or we can go click that and we'll download it for us. For these supported genomes, the gene positions, the GTF, are actually imported automatically. So we saw here, I got the ref genes automatically, and when I loaded MM10, I got some custom genomes here, so it's not the best example. Maybe C. elegans. I also get the genes there as well. We can actually import additional gene information into IGV using this GFF format. So if we wanted to, we can just put it in our own gene models. I'm not going to cover it, but I'm just going to show you that here now. So this is our drop down, and it's in here by default. Usually you have MM10. Yep, there it is. We have HT19. Usually it's got CE at 11. But if you don't see your genome of interest here, just go down to more, and it's got this full list of ones that it supports. Where we can see it's got various versions, including the new mouse, the MM39 I mentioned earlier on, but the MM10, MM8, MM9, the human. So if I wanted to work on this genome, I just press that. It knows to go download the genome for me. I think it changed. There we go. Got the GTF, and now I can start working on that genome. If you don't find a genome, you don't find your genome of interest in there, like we were saying, genomes are just FASTA files. If you can get that FASTA file, you can actually just push this in here, give it a unique identifier, a descriptive name, and then you can just start using IGV. Right? So you don't need to have it pre-built. IGV doesn't need to have it in their server. As long as you have that FASTA file, sequence information, for your genome build, you can just browse your computer, put it here, and it's fine to go. Again, you can add, we don't need to worry about cytobands, but we could add a GFF as well, and we'll get the genes. Once we've created this genome file, if you're using a strange genome, then it's going to appear in here, which is why I have lots of stuff on monkeys and something called E and chicken, and all this. We just built up some custom genomes. Okay, so once you've got your genome data and you want to start putting your actual genomics data, your high throughput sequencing data onto this, IGV is very good at or caters for the wide range of different data formats. I think I've probably got a link here. I can show you all the different data formats it covers. Documents, file formats, covers many formats, and lots of the ones we've described, math, bigwig, big bed, BAM, and lots we haven't even talked about. Vcf's there, STAM, probably Wiggle, Wig, all these different data types. IGV caters for most of them. We're going to find it's going to be, I think, tough to have a genomics data type which IGV is catering for. IGV also does allow for the inclusion of metadata information about your samples. So I said that that panel on the left can contain metadata information. It can do. By default, it's just going to contain the name of the file. But if you want to include more sample information, you can actually provide a file to IGV, and it just needs to follow this format where you have, and there's an example here, where you have the name you want for the file, the type of data, and then you can have additional metadata information here. And then you can use that in combination with bigwigs to load this up. Actually, we have a package we wrote in the BRC, which allows you to do this. I think we'll show this later on in the course. This is track tables. It's publicly available at Bioconductor, so it's a publicly available package. And it just talks you through how you can take a sample sheet, lists of bigwigs, etc., and create these HTML links and be able to provide IGV with that sample data information. With that, you can do additional things in IGV, and this is why we really like it, because it allows you to sort and filter your groups of samples. It can even allow you to actually split your tracks across particular groups. So you can go from, If you have multiple antibodies for a particular transcription factor, you can start to group it by these features, say here, antibody, or maybe even a species here. And it just makes it a lot easier to arrange your data. Again, I think rather than do that using this system here, and you can go and follow this here, I recommend, this isn't a programming day, so we're not going to go into it, but I recommend using track tables. Exactly the reason why we wrote it. Okay. So as with UCSC, so this is this web-based server, IGV does also allow you to support data, or it does also allow you to load data from external servers. So data accessible from a URL, such as an HTTP, HTTPS, or FTP, can be loaded straight into IGV using file load from URL. So I'm actually just going to load some data now. First, I'm going to copy this link. Copy link. So just to show you, IGV I think is really neat and quick at this. So let's load some data. I'm just going to load some local data. So there I went, file, load from file. Right, we got below it, load from URL. We'll come to there in a second. So I should have just downloaded some data for us. There it is. So bed file, I don't need to do anything different. I can just go open. I loaded the bed file. Like I said, it does actually put it in this bottom one for some reason. You can go file, load from file, or load a bigwig. Okay, it's going to put it up here. And again, the instant, right? And that's because that bigwig was binary, it's indexed. It's not actually showing us, I think this might be mouse data, which is why it looks so weird on there. Sorry, I'll start that again. You could tell it was mouse data because I was missing some chromosomes over there. Load from file, bigwig, we'll just load them all at the same time, maybe. So it's loaded the bigwig, it's loaded the bed, and we loaded actually here the BAM. It's just telling us something we can talk a bit more about later. It doesn't want to load in all the alignments in one go to try and visualize it because it'll be a little bit computationally expensive. So it's easy enough just to load from local files. We can just remove them now. Maybe I'll remove these ones for now. Remove tracks, easy enough to remove tracks. If I want to load from a URL, just need to specify the URL. Here I've got my HTTPS address. I can just load that. It'll take a second. There isn't much data there. There is a little bit of data just over this particular bit. Just a little bit of data. We'll also go load from server. I think that's the next thing we'll do in the class here. So like I was saying, UCSC, which is the web-based one, comes with loads of pre-built, pre-installed tracks. So if you want to look at, histone marks in a particular cell line, it's already built into UCSC. IGV, because it's running locally on your computer, you're not going to have that data available to you. But you can actually load in a lot of data to IGV using this load from server feature inside IGV. So I think I'm just going to show you a little bit of that. I just want to check what genomes we're working on here. So if we go in here, we can go file, load from server. Actually, I don't have much available for MM10. But if I change genome to HG19, I go file, load from server. We've got lots of information here. We've got lots of things we're going to dig up later on, but we've got regulatory transcription factors, 1,000 genomes information, histone marks from Broad. We just need to, I'm not going to actually load it because it will take a few seconds. It's going to load it over the internet. But IGV doesn't quite have the databases that UCSC does, but it does have something in way of servers that you have pre-computed annotation. Is it fun to load different types of files, bed, bigwig, and IGV when making a figure from IGV? Yeah, absolutely. I would hope so. Absolutely. You want your bed, you want your bigwig side by side. Absolutely. And then you can arrange them as you like. And we're going to get a bit more into that now as we go on. I'm just going to quickly turn off my video. I'm going to get my charger. I'm sorry. Back in a second. Sorry about that. Yeah, and this is even, I mean, this is what I just did, but you can also, and this is what I encourage you to do in your own work, because ENCODE has all this data available here, and we know that these formats are available or are accessible in IGV, you can just go here, click this, get all your data. And this is exactly what we just did. We just loaded that straight into IGV. Okay. Nice thing about, at least for HT19, and I'm sure IGV are going to work on this, is they also have behind the scenes there ways of loading in data straight from ENCODE. You can search this for your favorite chip, Nick. CH12, is that there? Look at that one. Peak, there's a big bed. I can load that straight in. There we go. Oh, and we got some of this metadata information I was talking about too. That looked terrible when I loaded that though, so maybe I'll clear that and get a new session. Okay. So I think we're going to come to the end of the time now. If you want to sign out, I've got it recorded. I'm going to be about 10 minutes now to finish the rest of the session. So IGV associates common file formats with default display methods. Okay, so actually just now, we'll do it again. I loaded a bed file, and that actually ties nicely to your question as well, Abbas, right? I loaded a bed file, a bigwig, and a BAM at the same time. And IGV knows to display BAMs and bigwigs and beds in different ways. Okay, so this is kind of dense, and I can get an idea of the signal across my chromosome. BAMs, it wants me to show me a coverage, junction, and the reads. I'm not zoomed in enough to see it yet. And bed looks a little bit. So IGV makes some default display choices for us. You can actually go look at these recommended formats here, and it will tell you what it's going to try and choose to do to your type of data you put in. So it's going to say, with a bigwig, I'm going to try and do this by default. Just to show you what they should look like. Your bed file, and we remember these are our very simple, often just chromosome start end. They could be bed 6, so they have an ID, maybe a score. They're just going to look like blocks in IGV. Right? They just look like this. Blocks giving the start and end, and in this case, some ID, some name. Wigs, or bed graphs, or bigwigs, which is the much more friendly way of doing this, and I don't encourage you, but if you ever want to compare memory use of a bed graph versus a bigwig, it's very, very different. These allow you to see, your profile of your signal. And in this case, I'm looking at ChIP-seq, and I could look across here and say, I seem to have a peak at this particular point. Maybe I have a binding site at this particular position in the genome. BAM files, they contain your alignment information, so they contain sequence information, where it aligned, how it aligned, all this. That's going to be complex to display, and actually IGV does a really good job. It's going to show you your coverage, right? So it's actually going to calculate the depth of reads at every position, and it's going to show you underneath every single read. So IGV does allow, we can actually just show you that briefly now. I've got it loaded here. So below, go in a little bit closer. Oh, I think this is probably on the wrong genome. So let me try that again. Genome 10. I just want to show you what BAM looks like there. So, oh no, wow. Don't need to load the BAI. I go into our, you can see here on the bottom, this is our bigwig of signal. Auto scale. Here, if I zoom in a bit here, these are actually my individual reads from my BAM file. So this contains the same information as I showed you when we looked at that BAM. And you can see we have a read name, and this is obviously on a machine called Colombo. Cool. It's on the flow cell 8, like we saw. Then we have the position within that flow cell 55, 1118, 1799. Then we have *0. I'm not quite sure what that is, but we have the \1, so we know it's the first read in a pair. Okay, and then you have those tags we talked about. So those additional tags, we have the location, I'm sure other useful information in there too. What's really cool is that IGV under the hood has also calculated the coverage. So if it piled up all these reads, this is actually what that looked like. So it's kind of calculating a bigwig for us as we go. So IGV allows you to really customize the display of these tracks. By default, it did all that for us. We can customize this display ourselves, either through view preferences or from select track. We just select the track on the left-hand side and we right-click it. First thing we're going to do is just look at the one on the top, which is our view preferences. So we're going to go through this. And I'm actually just going to step through it inside IGV rather than the slides. Just going to have a quick look to see what we have. So if we go view and preferences here, we have first the general preferences. So this includes, show the attribute track. We do want to show that. Use relative paths. The type of fonts we want. Okay. Really usefully is this feature flanking window. So when I zoom into something, how far should I be flanking around the region when I zoom into it? We have things like tracks. So this is the size of my tracks here. So if I can't actually click on the track, but the size of my track windows here, I can control using these. And then I think one which is most useful or super useful to a lot of people is the alignments section. So I said by default, it's going to show us the alignments, the coverage. It's also going to show us something called junctions, split reads across exons. If we wanted to, we might actually not care so much about seeing the reads. Maybe I just care about how they pile up, how deep they are. Maybe none of that, and I just want to see the reads. This is all going to save me memory in some way by just asking the computer to do a little bit less. Downsampling is something which I think a lot of people don't realize is sometimes happening in IGV. IGV, in order to save memory, is going to try and downsample some of your reads. Once it reaches a certain depth, I think it's 100 reads in the window, it's going to start removing some of your reads just so that you only see really digestible information. But various things on like whether or not we're going to filter duplicate reads, so reads exactly the same position, with reads which have low mapping quality, all sorts of things we can filter by. Worth pointing out that they're on by default here. And I think one which is really useful if you want to really review your BAM files is this visibility window. So just to show you this a bit. If I zoom out here, I can still see my reads. I can still see my reads. Actually, I'm quite far here. I'm 33 KB, 100 KB, 270 KB. I should now. There we go. So now it's gone. That is actually controlled in there by this view preferences, alignments, and then the size of the window that you're willing to let IGV calculate these alignments over. Let IGV visualize these alignments over. So if I make that a lot smaller, I'm going to have to zoom in. I would have to zoom in a lot further, right? I can't see it at the resolution of, it's now 60K. I actually have to go underneath that. I'm going to change that back because that's going to confuse me later on. The alignments, so on top of the top area, we can also do some nice cosmetic changes to our tracks. We can do things like change the color. It's kind of straightforward. We can change the type, right? Type of graph that we want. So by default, it was I think, bar chart. We can turn it into a line plot, a points plot, or even a heat map. I'll show you this again. Really cool thing and important thing in IGV is scaling your data. So we can actually ask it to set the maximum and minimum of the scale of the visible window. Slightly more complex to that is a feature called auto scaling. So auto scaling actually allows you to fit the window to the biggest part of data you're showing. So let me show that in action. So I've got a few things I wanted to do there. Let's zoom over here. This is a perfect place to show auto scaling. So I can rename, I can make this a different color. I can make all the positive values orange, for instance. I can change the type. So it is a bar chart by default. I can make it a line plot. I can log scale it. That's not particularly interesting to me. Or I can make it a heat map. I'm going to go back to bar chart. And then I was going to show you this auto scaling. So it's going to try and fit the window to the maximum size of the data. So if I auto scale, it is auto scaled now, you can see that these things look different sizes. If I shift along a bit, we can watch that this should get, let me turn auto scale back on. There you go. So that will get bigger. And actually the range of the window changed, right? So 0 to 932. I go along, this became a little bit bigger, right? So it's trying to fit so it can show us the available data. This can be really useful for finding patterns in data, right? So actually I want to see the pattern. I don't really necessarily care about the height, but it also can be very dangerous because you jump between your regions and you think they're at the same height. I want to turn it off, I can turn it off. And I could also set my actual data range here. I'm going to hard set it to 50 at this point. So final bits of the class now, some really nice, useful features. IGV, beyond just showing your alignments, it can actually show your splice junctions. So given that we know genes are stitched together from exons, if we have different transcript versions of genes, like we have here, this is one gene, we can actually identify this just from IGV already by looking at these splice junction changes. I've got a sashimi plot there as well, which is just another way of visualizing this in IGV. So I'm just going to load some data quickly. Load from file. Do you know what? I'm going to go back to human. If I change genome now, it's going to unload all my data. It's kind of a pain, but makes sense. Load from server. I'm going to load this tutorial quickly. I'll show you this one. Oh, I can't remember the gene now. SLC25A. Down. Let me see if I can spot the gene from here. I can't remember the gene. I have to come back to that. IGV allows you to see splicing effectively. It can calculate these splice junctions just from the reads themselves, and it will be able to create these images for you, which is actually very cool, because you would often have to use other programs in order to do this. In fact, there's a program called Miso, which is why this is called Shishimi. So IGV can do that straight out of the box. And I'll try and find the region. I think it's in our exercise, the region where we'll see this Shishimi splicing change. IGV allows you to overlay tracks. So this is very nice, and it was a feature in UCSC for a while. And again, we're going to cover this in our exercise, but it just means that if you have 3 tracks and they're all different colors, I can overlay them, and then that might allow me to see the change between the patterns. I might see the blue take over, this mark took over at this point, all on one track. So we can do that very easily just by using the overlay feature. And it's just if I take 2 tracks, let me try and load 2 tracks. Bigwig. Geez, we don't have had enough data today. Well, these all look like the same data. Let's try that. Well, we'll do it in the exercise anyway, rather than me hunting for data at the end of this. But you can just, as long as you make them different colors, you just select the tracks and do create overlay track, and it just does this nice overlay, and then you can start to see the differences. Very cool feature. See, here's Bed Tools. A very cool feature of IGV, and it's still there, is this tools combine data tracks. Okay. So actually IGV allows you to do what is called track calculus. So if I wanted to subtract the values of one bigwig from another, say 2 different conditions, I can just do that here, and it will calculate a new track for me containing that information. because they're the same track though, it's not going to give us a particularly useful example here. So we could do the sum of two tracks, you can do the division, you could do a log, transform them, and then do the division, create these new sum tracks. Another thing which is, I think, useful in IGV is these tools. If we go here, we have run IGV tools. IGV tools actually has some methods here to convert a bound file into what is called a TDF, which again, you can visualize in IGV like a bigwig. Okay, it also has tools here to allow you to index your bound files, so you can create indexes from your SAM to bound files. So it has a little bit of tool set inside to allow you to do some bioinformatics analysis. Personally, we're going to learn a little bit of that now using R, so I would recommend doing that. But if you wanted to, you could also delve a bit into IGV tools. And I think there's a description here about creating what these functions do and what they'll allow you to do with some of the files we produced. A little sad, not sad, but it's a little of a shame. TDF is an alternative to bigwig, but for whatever reasons, historically, bigwig kind of won that format war again. So even though IGV has a format proprietary to it, this TDF format, it's actually not used much elsewhere. Bigwig is way more common. IGV tools allows you to create this TDF. The real winner, I think, for bigwig is that bigwig we can import into R and manipulate later on. TDF, we just can't. So if you want to save images, and I think this is an important thing for people, they get to a point, file new session, we actually get a decent file here. If you want to save this image, you can do this in IGV quite easily. Load from file. Where do I get these from? Loads, this big would say, this big. You can just go file, zoom into the area, do your coloring as you require. File, save image. I think it was SVG, I believe. Like a vector-based format. And once you have that, you can actually take apart your image inside things like Inkscape, inside things like Illustrator, and remove tracks, change colors all yourself. So I know a lot of people use IGV for their final publication image. They just need to take it into something like Inkscape first to allow you to do a little bit of tidying up. Final thing I think we talk about here is once you've done all your analysis and you've loaded your data, or you've done all your visualization, you've got all your data colored and organized the way you want to, you might want to save that session so you can come back to it. You can do that here. Just need to do save session, name it something. And then save it in the downloads. It's going to save it as an XML, and then next time we can just load that XML, and it will load all the data you've had associated in that current session, and it will load the colors. So that's the end of our sessions for today. I'm really sorry that was a little long. I do actually have, it's not pointed here, but I do have some exercises if you want to run through them. Just one exercise, but it is good practice for using IGV. So feel free to work through that yourself. I will come back with just before 5, and I will step through this with you. One thing I notice is that IGV updates a little bit. If you run through these exercises and you get to this point, you might find that I think the CTCF and this one are no longer available. But don't worry, just load what you have, do the overlay tracks, and actually it's kind of a cool little exercise to investigate activity of genes and then splicing across this particular region. So it's not at the end of the course there, but it's in the exercises. Exercise 1, I guess there's a solution, but the solution is visualizing it. So I'll come back at just before 5 o'clock, and I'll just very quickly run through that. Okay, thanks a lot, guys. I think we had a question from someone. Someone wanted to talk a little offline, maybe. I think it was you, Carolina.
Oh, it was to download the gene, the reference genome. Yeah, when I click into it, like a white screen appears and then it pops off. So there's nothing that downloads, at least in my computer. I don't know.
So I know that like iGenomes, in my background, it did just pull up a white screen, but it download something as well. So I know I've had problems with iGenomes before with like older versions of stuff. So you just maybe just try getting like an update to your browser and just
Okay. I'm using Google Chrome, so I'll go to Safari and try.
Yeah, usually actually I think Google Chrome is pretty good. I think it's there was some security thing or something, and once I fixed that, but actually before that it wouldn't let me do anything on iGenomes. It's a very secure page.
Yeah, I don't know if I had to you know, log in from an institutional VPN or something, but
it should all be free. I don't think I'm logged in, so it should all just be free.
Oh, now it, okay, great. Yeah, it lets me download it from Safari, so good thing.
I do warn you, it's going to be a big download, I suspect.
All right, I have a big hard drive, so we'll see. Thank you so much.
Okay, take care. See you in a bit.
Yep.
Share my whole desktop. Okay, so here we have our questions on the right, our IGV on the left. Hopefully you can see it all. Am I recording again? Yeah. So load IGV, select the HG19 genome. Yep, I got that. Great. Navigate to this position. Nice thing about IGV is you can just copy and paste anything like that. So I can just hop to that position, and a lot of the time people write it out like that or without the I'll do the same thing. And actually there are nice tricks in Bioconductor that we can turn that into a kind of GRanges bed file. Navigate to gap.dh. Oh, there you go, it's right in the middle. Zoom out to see the surrounding genes. I can't quite zoom out here, so I need to expand that a bit. Zoom out. Okay, fair enough. Select the ref genes track. So if you want to select a track, it's always on the attribute panel on the left. Right click, then expand. Expand. So then we have lots of genes here. And I think hopefully one of them was called Scarna. So we had a missing A there, but it was Scarna. So if we zoom in on that a bit now. Okay, and then click on it to bring up a window with further information. So that's going to take me to a new window here. By the way, it's a nice feature of the tracks which are built into IGV at least. They're going to link you back to NCBI, and I can go in and get all the information on this gene, maybe what it does, and links to other sources of information on it. So it's kind of nice. We'll click straight to NCBI. What else do I have to do? Double click on the gap.dh. Okay, so I'm going to zoom out a bit really, and then double click on that. It should center it. I think my window, oh look, I can, maybe it's just zooming in on it. Right click the region of interest. Oh, add a region of interest. So I'm going to do this the other way, the way I didn't do in class. So I go region navigator. And if I want to make the whole region a region of interest, I can just go add, and my present window became a region of interest. If I zoom out a bit, it becomes more apparent. The other way to do it was to select, that would also give me a region of interest within a region of interest. What's happening here? My IGV is starting to die on me. Let's just concentrate on the way I did it. Okay, zoom. So that's gap DH. Edit description. So if I click here, I can edit the description and I can call that an active gene. And that's going to be useful if I bring up my region navigator, because that's actually going to have the description there. Okay, menu load from server. So we talked about this just at the end of class. We don't have all the data we used to have available in this tutorial, so we only have a few of them. I think Matt made a point that you could also have gone to encode, so you'll probably find it in encode. But I think this was good enough to work with. If we just click there, like I said, we just load those ones. It'll take a second, and they do load. Now we have our signal from at least the K27 acetylation, K27ME3, K36ME3, and the K4ME1. So we have some active. I think this is a repressive mark. So I think this is all of them. Active. Probably have to Google it if you don't know. I think it's active, 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 repressive. Select all the tracks and go auto scale. Okay, so it's going to put them all on their own scale. That's on a different scale to that, to that, to that. And chips, actually, that makes sense. Chip have different efficiencies, so you would compare the same mark maybe under the same scale, but this is different antibodies, different experiments, so they may be on different scales. Go back to the gap DH locus, zoom out a bit. And really, we can see we have this K4ME kind of over the body. We definitely have the K36ME3 over the body, right, gene. And then we have a lot of K27 acetylation at the start. It's even clearer if you zoom out a bit. Okay. Okay, so let's try a different type of gene. Select all the tracks to maintain current data ranges. Okay, so we'll do that. So it's no longer going to auto scale, and I will go to PM AP. It's a very different looking gene. In fact, now we have much higher K27ME3. Right? So if that is the repressive mark, then this gene is definitely repressed. Edit description. So I need to add a region of interest. Let's try it again this other way. See if this works. Worked. Now I can. Sorry, Mark. I got started a little earlier, but I'll have the recording to for you to look at as well. So we can edit the description here. I can add in for this inactive. Okay, so now I have two regions in my regions of interest. I got my active, and I can zoom to that. We can see that that has a very different pattern. High at K27, high K36ME3, also a high K4ME1 here, but very low K27ME3. If we skip to this other one, very different, very high, off the chart K27ME3 here, barely any K27 acetylation. So that's an inactive gene. Zoom out to compare enrichment across neighboring genes. Well, actually, these genes look kind of active, if that is activity, right? That looks very different to that gene. We'll put them all on auto scale again. Yeah, I mean, this gene and this gene are day and night, I think, really, in terms of this mark at the promoter and this over here, and then this seems to be mostly K27ME3. So that's probably what we wanted to do there. If we can zoom out a bit more, maybe there's another repressed gene. That they are very opposing, right? Actually, we can just. So they're very opposite each other, this mark and this mark for sure. Go to regions, gene lists. Oh, great. So we get to go see our pro-neuronal dev genes. I mean, what are we looking at here? In terms, oh, sorry. View. Now we can see actually a lot of these look repressed. Right? Maybe this one's active. This one looks kind of repressed, or at least that. This one looks kind of interesting. If we zoom in. Yeah, that's very interesting. So maybe this gene here, TCF, this is one of the proneuronal genes. Maybe we have different splice of different variant here. So this, if we expand the ref-seq genes, this looks kind of repressed. It looks a little high over here, but this lined up with these other transcript variants. So maybe this versions of the transcript are expressed, whereas these ones don't seem to be. There is one little mark here which seems to line up with that as well. Okay. Select the signal across the genes to determine their expression state. We kind of did that. Return to main view by selecting all from the chromosome. Color all tracks by unique color. Okay, yeah, that's a good idea. So this one's not a unique color, so let's give that something really opposing. Let's do something cold would be nice, blue. We can make that purple maybe. Something brighter. Okay. Actually, it looks a little. Anyway, let's not go too much on the colors. Now, with all of them, we're gonna do an overlay track. So I just need to select them all and press overlay. Now I have my overlay track. Change the track height to 100. Hundreds. Oh no, track height. Change track height to 100. So this one. Okay, not the scale. And then revisit gap DH. Yeah, let's see that now. Okay, so actually that looks kind of useful, right? The other marks don't really matter. I see a lot of signal here. See a little of that yellowy color there. And then I can get a PAMP. It looks very different just from this overlay. And then I can do that neighboring gene thing again, right? Okay, very cool. So that's the end of that, and you could, obviously continue and scanning across. But I hope it shows you that, using public data, you can get an understanding of, in this case, the state of all these gene expression, the state of all these genes. Are they expressed? Are they repressed? And this is just ENCODE data from the tutorial, but as we know, a ton of ENCODE data available to us. So if you have a favorite gene under a favorite condition, it's worth looking at ENCODE and seeing if you've got something you can pull out straight away. Maybe review your hypothesis without having to do any experiments yourself. The other side of this is RNA-seq. So let's start a new session. And then we'll do load from server, and we'll take the tutorials. Oh no, we'll do the other one, the RNA-seq. Select that. Okay, and then I got to go to tutorials, view preferences, alignments. I'm going to change my range. I'm going to be able to look at, and that's going to show junction track. It's already on. Set visibility range to 15K. It's way higher, but I'll do 15K. And go to the SSL gene. So save that, and I'll go to SLC 25A3. Collapse read tracks named heart. Collapse reads track. Okay, so I'm going to collapse this. So named heart or liver. Okay. Select the tracks, color by alignments, color alignments, read strands to identify strand of transcript. So I can already start to look at the alignments and start to get some information here. So read strands, color alignments by read strand. That was group alignments by, sorry. Color alignments by read strand. Jeez. There we go. actually, it looks stranded. It doesn't look stranded there, to be honest. So I'm not sure what the intention of that was. Expand feature tracks to identify exons transcript. So if we collapse this, we can, this is what it looks like, and it gives you what is a representative transcript. So you can see all the exons which could occur in a gene, but you're not really seeing the transcript model. So if we expand this, we can now see the different transcripts which potentially could come from this gene. And we got one here, which is very different from these other two in that it has an exon here, but skips this exon. And it's really that we're most interested in this particular point. And inspect the junction tracks to discover areas of coverage unique to heart and liver. If we look here at the heart, we have a really clear coverage there over this exon. Liver, we have a really clear coverage here over this exon, and it's almost absent here. There's a little bit of it here. If we zoom in, so here, what we have here is a really good example of a splicing event. Okay. This exon is used in heart, but this exon is used in liver. Okay. What we might also then want to do, and it says here, is look at the junction track. Oh, sorry, gone too far out. Zoom in a bit. We can actually see also we've got evidence. It's not just reads mapping here. We've got evidence of reads mapping from here across this to this exon. And these are called junction counts. Now this is good evidence that this really is a true exon. And this splicing seems to be happening between these two conditions. We can expand this if we want to inspect the different splicing kind of events happening. And you can see that these ones go from this exon to this exon. And there is some minor exon there, it's still there, but this is the one going from there to there. So we can look at both the coverage, but also the splicing of this mapping of a read starting here and ending here. Junction to identify, yep. Okay, so if we click on this or if we look over it, we can see where it starts and where it flanks to. Something which is really nice in IGV is this sashimi plot. So if I just right click on a junctions track here and I go sashimi plot, and I select the samples I want to look at, I go okay. I get this plot, which is originally derived in some other software, Miso, but it's great that I can actually see now some quantitation of this. I got 726 reads mapping from this exon to this exon. I have much less in here, but I have 2,836 mapping from this exon to this exon. So it starts to give you some kind of numbers to tie to this actual shift, this change. Okay, select gene track. We did that, and then we just save this image, right? I think it was this one, select gene track. Select that. And I think the last thing we wanted to do is just go File, Save Image. Oop. I got an alarm beeping at me in the background too. Oh, sorry. Actually, most off, you're going to want to save that not as a PNG. You're going to want to save that as a SVG. So you can save that. Okay, so that's the end of the session for today. We have no session on Friday. It is a long weekend from bioinformatics. It is for the break. Have a great 4th of July, and I will see you all after the break. If you have any questions in between, hang around now if you have anything to talk to me about now. Or we will catch up after. Oops. Hey, Matt, I'm going to go. I'll speak to you soon.